Hello everyone, myself Vijay, and today we are discussing about the topic coordinative properties of the enzyme. Now we discuss about the cooperative properties of, of enzymes. Cooperative properties can be of two types: positive cooperative property and the negative cooperative property. First of all, we will see what is the enzyme. These act as a catalyst, or they act to convert the any substrate to into the product. And they present in the body. Now we will see the structure. Like example, we have enzyme. Okay, now we will have like a active site here. Active site. At this site, substrate binds to form product. Now we will see the reaction. Like we have a free enzyme here. Enzyme also have the active site here. Now we will have a substrate, blue color substrate we are taking here, denoted by S. After that, they form a complex, like enzyme substrate complex, like this one. This is known as the complex. After that, enzyme get detaches and form the new substance, or we can say the product. This is the product. This is the enzyme. Now we will talk about the positive cooperative property. Let's take an example. We have enzyme and it have four subunits. As substrate came and bind to the first subunit, and now it's change its shape like that way. All the other three subunit are of same in structure. Here the substrate. Now all the three subunit like ask, or we can say coordinate with the help of chemicals and other things with the first subunit which get attached to the substrate. and changes its shape, and after that they also want to change their shape. If they want to change their shape, and if they change shape, then we can convert more and more substrate to the product. So it is the positive affinity, positive cooperativity, or the affinity we can say. In this first one, uh, enzyme with four subunits, all subunit are same, like all the four subunit have the same active site. Now here there is the change in structure, also known as the conformation change. Okay, now we will talk about positive affinity. What is their effect? Like, first of all, have low km value and high affinity, because the like other subunits want to like change their shape, so we the substrate can easily attaches to it, so affinity is high affinity towards substrate. Okay, now we will see about the negative affinity and their graph. First of all, negative affinity, or we can say cooperativity. Now what we understand with the negative cooperativity, let's take another example of an enzyme have four subunit having the same subunits. Enzyme have four same subunit. Okay, now and substrate came here, like box like shape. uh, this is the substrate, and if the substrate came here, there is the conformation change, like it changes its shape. Okay, but in this case, when the other three subunits don't want to change their shape, don't want to change shape, so there will be a negative cooperativity. If it is negative cooperativity, then it have high km value and low affinity towards the substrate. Okay, now we will see a graph here is the rate denoted by V, and here is the substrate concentration. This type of box we are seeing in this video are used for the concentration. Okay, now when we see about the red color denotes the positive affinity. First of all, the substrate is coming in low amount, but when all the subunit changes its shape, then it start increasing the substrate into the product, and the rate in the first of all it is low. After that it increases, and then the saturation why only like we have four subunits, they can change four substrate into four product, so there is the saturation. If we have like infinity number of uh, subunits or enzyme and infinity number of substrate, then we have infinity number of the products, and then the graph get changed. But in this case, this is the positive affinity, and it is of sigmoid shape. And when we go for the negative affinity, like firstly it increases due to some of the substrate get attached, but after that words it get decreases because the other do not want to change their like shape, or want to in the conformation change, so it's decreases. Negative cooperativity graph. That's all for this video. Thank you. And now we will also have another topic of hemoglobin and myoglobin. Some of the points I want to discuss about them. They are not, first of all, they are not enzymes. Second one, they like work on the reversible binding property or the function with the O2. Myoglobin serve as to store O2 for its consequent utilization, like a whale and a human. If we compare, then the whale have high myoglobin as compared to the human because they want more myoglobin, because it serve as a storehouse for the O2. And on the another hand, we have hemoglobin function as functional protein of RBC, uh, services for the transport of O2 from the lungs to the tissue or the cell level, and then carry the CO2 from there and get converted into out from the body from the lungs. O2 can be like we talk about the shape. If we talk about the myoglobin and hemoglobin shape, there are of crystalline shape. Hemoglobin have four subunits: two alpha and two beta. Similar but not identical. They are similar but not identical. After that, when we see in the hemoglobin, there is a heme uh, like bonding or configuration. They also contain the iron atoms like Fe2 positive, and more lots about this we discuss. And thank you, and have a nice day.